I'll record on my computer.
Okay.
Okay, now we're starting. Okay, so we did a couple of them right now. We talked. I was doing a couple of the stuff independently from that recent PowerPoint. Let me see. I talked about the light reactions or something. It was like the light cycles, light response curve. I noticed from the PowerPoint there's a couple good important stuff. I think the best way, we just run it down from each, of Dr. Tobin's voice threads, and then, write some sort of straightforward response, like simple response for us to remember.
So we fill out those contents.
Exactly. Like I converted this PowerPoint or PDF to like a little Word doc here, and then we're just gonna fill out little stuff here and there.
Yeah, that's cool.
Awesome. Let's see.
I'll just do it like how I did it in Cancer BIOL then.
Exactly, same thing.
Looks like you didn't do the embryogenesis yet, so I think I can go ahead and plow through that.
Yeah, that's fine. Yeah, we can start from the beginning there and then on.
Let me get my notes.
Let me fill this out here. Let me see.
Okay, so I'm gonna define these three. Embryogenesis.
I'm gonna refer to the PowerPoint in a moment. Let me refer to the PowerPoints. Give me a sec. Let's see.
Oh, I tried to define embryogenesis, but I think like there's already the answer right here.
Oh yeah, I have it right here. Sorry.
Let me just move this.
No worries. You can.
I guess I'll just add the seed maturation then.
Yes, you can put it over here. I have a little subsection.
Oh, right there?
Yeah, no worries. I'll handle the germination. Let me just get the PowerPoint to refresh my memory here. Let's see. How do you feel about exam 2 or exam 1?
Exam 1, I did very well. Like
Great.
I did have a few hiccups with the multiple choice, but otherwise the video, perfect score for that one.
Nice. Congrats.
But multiple choice, I did have a few questions where I messed up.
Same here. Yeah, for the video, I got like 17 out of 20 there. It really helped out. Yeah, the points. Let's see.
Seed maturation, seed develops.
Resumption of growth. So it continues growing. Resume. Let's see. This exam has a lot of, how do you say, like bio 1 review. I see a lot of it.
Yeah.
Calvin cycle and the photosynthesis formula. Yeah. Let's see.
Especially the lab, like when we learned some things like classifying fruits, it all came like back to my brain.
Exactly.
For the seed maturation, should I also like talk about the two different seeds? Oh, you got
Yeah,
it.
quiescent versus dormant. Yeah. Yeah, I was just looking at the PowerPoint. I remember quiescent, but I didn't remember the dormant part. There. Won't even germinate. That's the main difference. Looks like it. Let's see. One can germinate in optimal conditions of the dormants.
They need like an additional trigger, I think.
Yeah. Little water like a catalyst, but a catalyst is required. Yeah, like I did a couple back on Thursday with a couple others, I think. But it was kind of a long day. Yeah, I won't lie. It's week 7. We're almost halfway done. It's getting
Yeah.
tougher. Yeah. Let's see.
I'm assuming like other people, they can fill this out if they have time, right?
Exactly. I put it share. Everyone can be an editor. I try to ask for their emails too, but I haven't got anyone else at the moment.
That's okay.
But yeah.
Development of apical basal pattern.
Oh, sorry, I was talking about a little brother a moment ago. Let's see.
Okay.
All right, cool. Thanks.
Do you have like any PowerPoint for these two questions? Development of the apical basal pattern.
I think so. Let me see. It should be this one. Dr. Tobin's. Let me look at it. Let me go back. Apical, right? The
Apical basal pattern.
apical basal pattern. I
Did
should,
he say anything about that?
I think he did. Let me see.
Oh wait, it's along the
Right in the beginning.
main axis.
Okay. So apical meristem. So is this what we need here? Let me see. Primary.
I don't know if I should talk about the radial pattern. I'm not sure it's not going to appear on the exam.
I wouldn't be surprised it would, but I think just the development process of this apical basal pattern should be enough there. Let's see.
Okay. Because all I have on my notes is just says apical basal pattern along the main axis.
Oh, it's kind of big. I see.
Yeah.
Okay.
I do have the radial pattern. That's not in the study guide here, but I wrote concentrically arranged tissue systems.
Upstairs. Sorry, give me a moment. right, sorry, I'm back now. Okay,
Welcome back.
thank you. I appreciate it. It's my parents. They're trying to go to church or something. They're
Mhm.
kind of rushing. All right. So for the, are we still talking about the pickle and basil pattern?
Yeah, I only got the vague part of it.
Let me see. It should be still in the first one with embryogenesis.
Mhm.
Let me look at it. Let me see.
I think it would be more helpful if he had like a transcript or something of his
I
PowerPoint.
agree. Exactly.
Because sometimes I just get lost in like what he's saying
Mm-hmm. Same here.
and trying to take notes about like word for word about like what he's talking about.
Exactly. Like at one point I have to like slow it down or like just rewind it for me to like,
Mhm.
okay, this is what this means here. Let me see. Embryogenesis. Get it. Okay, I see what you mean. Yeah, pickle basic pattern along main axis. Okay, that doesn't really mean anything. Say anything there.
Okay, so just that
Yeah, yeah. I
part.
think that would be it. Yeah, along the main axis.
So for the next part, like, how do you think we should describe it? Is it just the basic structures, or
I think
I
so.
don't
Just the
know?
basic structures there.
Okay.
I
Um,
think. Let me see. That's where I saw in chapter in the second PowerPoint structure of the development. Well, I guess like what it consists of. I think I saw it
mhm.
over here. Like had like the protoderm and the over here, yeah, the primary meristem. So it's protoderm, the ground meristem, and the procambium. Let's see. Apical meristem. Let's see.
Second. Oops, no, that's not it. Apical.
Let me find it here. Let's see. Let's see.
I'm using Google. Not sure it's gonna be accurate to like what he's saying.
True,
And
it's
so,
okay. It's something. Yeah, no worries. Let's see.
so it's located below the root cat. I don't know. He talked about that in the PowerPoint.
I think you may have talked about it. Yeah, probably in this section.
Can you like double check about that?
Yeah, I can double check. Let me see.
Entire process of embryogenesis. Not only on angiosperms, but on eudicots. So we see that it all begins with the fertilized egg, the zygote,
Hmm.
then begins to divide by mitosis to begin developing into a mature sporophyte. It goes through this step here where there are only two cells, two-cell embryo, more cell division, eventually end up at the globular stage, then the heart stage next, and then finally the evocatively named torpedo stage. What we will do now is go through each of these steps individually.
Hmm. I don't see it here. Let me see. Two cell embryo.
Just
I don't know if this is even supposed to be part of embryogenesis. Weird. Let's see. Primary meristems. Okay, we got that one.
I'm gonna pull up my textbook.
All right.
I know. I'll go look at it.
Sounds good. Let me see. I'll pull it up. Let's see. Where is it? Pull out mine as well, just in case. Let's see.
This is an graphic that shows descendants of the apical or terminal cell.
Okay, apical cell. All right.
And the descendants of the basal cell in pink. Remember that the apical cell or terminal cell, as we discussed it, was the cell that was the result of that first mitotic division of the zygote and was closer to the chalazal end of the embryo sac, whereas the basal cell was the cell that was closer to the micropylar end of the embryo sac. Really see clearly here that the terminal cell or apical cell is the one that contributes most of the cells to the final mature embryo.
Okay, doesn't really talk about it here. It just talks about like how the apical cell pattern and the basal pattern, I guess.
I think that will be important.
That would be.
Do you want to add that?
I can. Let's see.
Okay, let me just move out the way.
No worries. I'll do another point. Let's see. Let me look at this.
Result of that first mitotic division of the zygote and was closer to the chalazal end of the embryo sac, whereas the basal cell was the cell that was closer to the micropylar end of the embryo sac. Really see here that the terminal cell or apical cell is the one that contributes most of the cells to the final mature embryo.
We see micropylar sac.
Micropylar end. Really clearly here that the terminal cell or apical cell is the one that contributes most of the cells to the final mature embryo. Remember that the apical cell or terminal cell, as we discussed it, was the cell that was the result of that first mitotic division of the zygote and closer to the chalazal end of the embryo sac, whereas the basal cell.
Chalazal? Let's see. Of the embryo sac.
Really see clearly here that terminal cell or apical cell is
Chapter 23. Apical meristems. I found it, Russell.
Awesome. Let's see. Tips of the root. Nice.
I'll just put the function because I feel like that can be a potential question.
That's true.
Now I need to talk about the structure. So I got the location,
Mm.
function. Mm,
I'm gonna go look and talk about more of the tissue systems on the next PowerPoint.
mhm. I think that's good enough, Russell. I
Mhm.
talk about what is it made of, and then
Christina,
it gives rise to the primary meristem.
that makes sense.
Do you remember that flow chart he showed us in the PowerPoint?
I do recall. Yes.
Yeah, that was the
Overview.
textbook.
Okay. Yeah, I'll make sure to look at it and review it.
Mhm.
Let's see. Do you think we should add like a picture or something?
If you can squeeze it somehow.
Okay. Okie dokie. Let me see.
If not, I can try doing a
I like a
snippet.
little drawing. Yeah. Let's see.
Oh, I got it.
Awesome. Okay.
Okay, so we knocked that out.
Awesome.
Now we need to see which one we had to do next.
Let's see. So I think we got the seed maturation and germination already set. Along with the dormancy factors, I did like a couple, like proper watering, sunlight, optimal temperature.
For the pictures of for the parts of the flower, do you think I should insert like a diagram?
I think so. I think I have I found a pretty good diagram here from Dr. Tobin. I can snip this and attach it here.
Okay.
Let's see. Alrighty, thank you.
Root system.
So we were going to talk about the 3 tissue systems and the root system. Let's see. So 3.
Yeah, I'll take the root
Okay.
system.
Alrighty, sounds good.
I have my notes ready.
Awesome, great. So we have dermal tissue. I'm gonna.
So root system structure.
System, tissue system. We covered a lot in just 2 weeks. I was so surprised, like exam was really quick.
Right.
Came really fast. Let's see. Let me see.
I should probably talk about the root types because there's 2 of them. Oh.
Internal tissue system. Okay. Huh?
Okay, got that out.
Awesome.
I hope those are good notes. I'll see if I can find a diagram to insert.
Alrighty.
Zooming out.
Yeah, looks good here. Let's see.
System structure.
Skin of the plant, dermal skin. The outer covering tissue or the skin of the plant, epidermis or the periderm. Let's see. I think that's straight to the point.
Yes, that's correct.
Mm-hmm.
Sure.
Okay, so grand tissue system, also known as fundamental tissue system. These are all the other tissues aside from the epidermis and vascular tissue systems. I guess that makes sense there. Let's see.
The vascular tissue system, isn't that the one with the xylem and phloem?
Yes, that's right. I think I remember seeing it. I was reading it there.
Mhm.
Vascular system. Yeah, that's right. Yeah, secondary phloem and secondary xylem, they divide. Let's see. Yeah, let's see.
I still need to do the voice thread for the
It's all right. No worries.
class. I hadn't started. I only read the book so far.
That's good. That's a good foundation.
Mhm. Have you started any?
Oh yes, I was able to finish commenting on the PowerPoints. For the quiz, I'm gonna retake it again because I got a 1.5 out of 2. I'm just gonna, gotta make every point count, right?
Yeah,
Yeah,
free 2 points.
exactly. It's not that much, but it's something. Let's see. And like I took Dr. Tobin's advice when he said, you take notes. So like when you're writing or like speaking, so when he's speaking or something. So like I was writing it a bit, and then after I would like put it into this study guide or the whatever study notes that he provided there.
Mhm.
Yeah, it really helped. Yeah, it's
I'm
kind of long
just
piece.
I'm just going in order of like when stuff are due. So I know
Yeah.
cancer bios due first, so I'm trying to knock that thing out
Exactly.
first.
The discussions, yeah.
But I'll have to like get some plant bio in today for the lecture.
That's
'Cause
fair.
I haven't started.
No worries. Yeah, I'm gonna switch to cancer bio after this. Do some per useful readings and all that. Let's see.
Is
I
it a
got
lot for this week
I
for
don't
the plant bio?
plant bio, not really. Aside from filling out the study guide and that weekly quiz, not that much.
Okay.
Yeah. But cancer bio, man, there's always so much stuff to do, Christina. I'm not gonna lie.
Mhm.
But I mean, like it helps us, reinforce like what we're learning. So I try to always remember that one, manage it. Let's see. Yeah, looks good here. The 3 tissue systems, xylem, phloem.
Hopefully, I knock out plant bio quickly.
Same. Like we won't stay here for this long. We'll do more independent work, and then we can probably. I'm hoping we can meet up again on the day prior to when the exam starts. So Wednesday, something like maybe 4 p.m. Something. I'm hoping we're having like some time, some space between classes, and like for people in the lab if they want to join.
Mm-hmm.
But yeah, but most of the time we're gonna just do independent work, just review it. I'm hoping to make a Quizlet before the exam starts, but it's a lot of content. Let's see.
Right.
Yeah. All right, this looks good. Okay. right, I'll continue the phytomeria here. Let's see.
Look at the development of the primary plant body.
The plants, okay, yeah. That makes sense. Okay.
Development of the primary plant body. Isn't that what we just went over? Primary root system, primary.
Oh, yes.
Okay, I'll just say refer to. Okay.
Looks good. Yeah, looks good here. Let's see. Auxiliary butter. I think this is an auxiliary. Let's see, describing buds or branches arising in the axil of the leaf. I got you. Let's see. I'll type it out here. Internode. Yeah, the part between two nodes, yeah. Part between two nodes. I think we're coming along pretty nicely. I think
Mhm.
like around halfway.
Yeah, it's a lot.
Yeah, it's a lot of content. We don't have to do it all in one day. We can spread it out for Wednesday, and we can do a couple independently. But I
Mhm.
think just referring to the PowerPoints and then the textbooks, I think we should be okay.
Yeah,
I think this is a good stopping point for now,
I agree.
Christina. Yeah.
Mhm.
And then we'll continue more independently and come back hopefully on Wednesday with it, whoever wants to join in. But yeah, thank you, Christina. I really appreciate that.
Mhm.
Let's see. I'll stop the recording.